Thank you.
Oh, the title of his talk is, it's sex, and we were supposed to do this, right? You're supposed to say the title. Sex differences in striatal neurons. Of course, all of us can read here, so I also have to hit this. Great. Okay.
Thank you, Chris. It was a very kind introduction, and I want to bring the expectation is high right now, so I want to bring it just way down, okay? And when you hear somebody talk about all this stuff, you certainly don't do it alone. I have a wonderful laboratory and many, many collaborators who have helped us not only make scientific achievement possible, but to really broaden it across multiple levels of society. I grew up in rural coastal Texas, and I had never even met a neuroscientist until I went to college. I didn't even know it was something that you could do. And I was very lucky that I just happened to fall into this. I happened to meet the right person at the right time, and here I am 19 years later having the honor of being a part of this life science seminar series. So I'm really grateful for not just the introduction, but also the invitation to share our work with you today. And indeed, I am going to spend quite a bit of time talking about sex and striatal neurons. And so just to really sort of place this in context, so Chris mentioned all the different phases of the things that I've worked on. And really what unites all those things across all the different animals and the techniques and everything else is that I'm really interested in just naturally occurring variations in neuron function, okay? So how do you take a neuron and have it be tweaked just a little bit so I can do something different, okay? And right now, most of our work is within the context of steroid sex hormones. And in sex differences, you're going to get introduced to my favorite molecule, 17-beta estradiol, and especially how it interacts with neurons in the context of biological sex to both change and not change neural properties. And so you could really distill my research program down to something very simple, mainly sex hormones in the brain, right? Sometimes you have rock and roll stars, they study sex drugs in the brain. I do sex hormones in the brain, okay? There's my first dad joke. I was talking with somebody about that earlier. Told you. So sex hormones in the brain, so it's very, very simple. And so one of the things that I've learned over the last few years is that we really need to take a moment and think about what is biological sex, because it's one of these things that seems very simple, but is actually a multifaceted experience, okay? So when we talk about biological sex, we can talk about different levels of sexuality. We can talk about genetic sex, which is things like chromosomes, right? So XX versus XY, at least in humans and rodents and primates and things like that. We can talk about gonadal sex, meaning mostly internal reproductive systems. So that's things like, does it have a testes? Does it have an ovary? Something like that. We can talk about phenotypic sex, which is things like external genital, secondary sexual characteristics and behavior. And we can talk about sexual or gender identity, which is an individual's perception of their sex, which also has a very strong sociocultural influence, of course, right? And so today, all the data I'm going to be showing you is from rats, right? And those rats, the rats that I'm working with, happen to have their genetic sex, their gonadal sex, and their phenotypic sex all aligned, right? But that is not necessarily the case, right? And biology has a wonderful variation, right? And so, of course, you can have these things be aligned or not aligned to create all the rich variation that we see in animal behavior of all sorts, right? But today, we're going to be mostly working with rats that have their genetic sex, their gonadal sex, and their phenotypic sex aligned. And so just to make sure we're all on the same page, I've talked about steroid sex hormones several times already. And so I'll remind you that steroid sex hormones are called steroids because of their ring structure, characteristic ring structure. Here, they're called sex because their initial discovery was in the context of sex systems, okay, and hormones because they're a signal that can be released in one part of the brain or in one part of the body and then circulate through the body to act on other things, okay? But I'll remind you that, for example, if we consider testosterone, testosterone can be produced in the testes and it can indeed circulate through the body to act on other tissues. It can also be made in the brain. Okay, also be made in the brain. So you can have neural source testosterone, you can have gonad source testosterone, other places as well. You can also have my favorite molecule. That's probably going to be on the test later, right? It's my understanding people have to take this for a quiz. What is Dr. Meitzen's favorite hormone, right? 17-beta estradiol. So 17-beta estradiol can be created from testosterone via the enzyme aromatase, right? And that can happen, of course, in the ovaries and circulate through the body. It can also happen in the brain of both males and females. And then testosterone can also be converted via 5-alpha reductase into DHT or dihydrotestosterone, okay, which is yet another hormone. And the way that we categorize these hormones is based off of the type of receptors that they bind to. So an estrogen, like 17-beta estradiol, binds to estrogen receptors. And there's lots of different types of estrogen receptors, okay? And I'm totally gonna gloss over that today and just say estrogen receptors. And then in terms of testosterone and dihydrotestosterone, they bind to androgen receptors, right? And just how in terms of neurotransmitters and hormones, whether it's in a plant or anything, right, those receptors help determine the action of that hormone. And depending on receptor expression and the different modulatory factors that can happen there, you can create a rich array of effects from a single hormone, okay? So if we take this back down again, back to the nervous system, historically, the role of steroid sex hormones in biological sex were primarily studied in brain regions that were directly involved in sex-specific behaviors. Okay, this is stuff like copulatory behavior, for example, or song in birds. Okay, directly involved in sex-specific behaviors with big sex differences in the neuroanatomy, like volume size of different brain regions, and in the context of brain regions that expressed only one particular type of estrogen receptor, a nuclear receptor. Okay? And so one example of one of these brain regions is the sexually dimorphic nucleus of the preoptic area, or the SDN. Okay? And this has been studied in rats, it's been studied in humans, it's there in some mice strains and not others, it's in lots and lots of different animals. And one of the things that was found about the sexually dimorphic nucleus, I mean, here's a very nice image from Gorski's paper in 1980, which initially described this, is that it's much bigger in males compared to females, which makes sense because this region is mostly involved in male copulatory behavior, right? And we know this is true because a few years later, many years later, Jordan Wong in my lab, one of my undergraduates, went back in and assessed the volume of the SDN in our rat population as part of another project, and we see this very nice increase in volume in male compared to female, right? So big sex differences, a clear link to behavior, and with nuclear estrogen receptors. And so these historical models were very, very useful, okay? And the reason why they were useful is that these robust differences that were easy to measure allow the elucidation of many basic principles of neuroendocrinology and sexual differentiation. And so one of these principles is that the neuroendocrine environment in which the brain operates is both dynamic, which means that it changes across development and in time, and that it's different by sex compared to males versus females. So for example, if we just consider testosterone exposure in male rats, here's conception, right? Here's when the fetus has a little surge in testosterone. Then right around birth, there's another surge in testosterone. And then things are quiet until puberty happens. Everybody went through puberty, right? And you know that stuff happens in puberty, right? Those testosterone levels went right up and things changed again, right? So this pattern changes across time, across development. And if we consider, say, female rats, right? In this case, estradiol, things are pretty quiet all the way through birth. And then in puberty, the cycles of estradiol exposure start up, right? A cyclical cycle. So that's a 4 to 5 day cycle in rats. It's called the estrous cycle. We're gonna return to that later in the talk. And of course, in humans, it's more of a 28 to 30 day cycle, but you also have cyclical changes in estrogen and progesterone and other hormones as well, okay? So these hormones, which are dynamic and differ by sex, can really do two broad things, okay? One thing that they can do is they can have a temporary activational change. And what that means is that a hormone comes into the system and it changes the neuron, and then when the hormone leaves, the neuron changes back, okay? So it's a temporary change. And the other broad category is permanent or organizational changes. And what that means is that the hormone comes in and it changes the neuron, and the hormone leaves, but the neuron is still changed, okay? And so this permanent and organizational change only occurs during specific sensitive periods in that animal's lifespan, okay? And so if we consider a rodent, like a rat or a mouse, there's one sensitive period that's right around birth and just before birth, right? Because you'll remember that in males, that's when that testosterone comes up, right? And that testosterone can maybe get converted to estrogens and other hormones, and they can change that neural system. And then the testosterone leaves, but the neural substrate remains masculinized and defeminized, right? Others, particular sensitive periods occur in and around puberty. Cheryl Sisk and others have done beautiful work with that. And then there are some scientists who argue that there's another sensitive period in and around reproductive senescence. These sensitive periods are also present in humans. They are not as well studied for obvious reasons, but we know that there's a sensitive period which the fetus is exposed to when masculinization can happen, okay? So organizational changes can happen, but the hormone has to show up during a particular time, okay? And if the hormone shows up in a particular time, so for example with the males, right? So if you have the XY, and on the Y chromosome there's a gene called SRY, which in conjunction with lots of other genes causes testes to be formed, right? And then in and around birth, those testes can secrete testosterone, which can get converted into estradiol in the brain and other places, and then that can masculinize and defeminize that tissue, right? And so this is a concept called sexual differentiation, which is just a fancy word that means how hormones in conjunction with other influences sculpt our neural substrate to produce a more masculine or a more feminine phenotype, okay? So all of the data that I just showed you, I'm able to show you because people spent decades and decades working on those classical models to produce those fundamental findings, okay? But there is nothing in life which is all good or all bad, right? We work in these sort of varying shades of gray. And I would argue that one of the downsides of the classical model is that they also help propagate an assumption, okay? And that assumption is that sex differences only apply to brain regions and behaviors directly related to reproduction, that they only applied to brain regions that had changes in gross anatomy and volume like the SDN, and that they only apply to regions that had nuclear receptors for steroid sex hormones, okay? And we now know there has been an explosion of research over the last, let's say 10 or 15 years, generally speaking. People argue about exactly when. But there's been an explosion of research that shows that that assumption is incomplete, okay, or not correct. And so we now know that there are sex differences in gene expression and splicing across multiple brain regions. We know that there's sex differences in the connectome, which is how neurons connect to each other. We know that there's sex differences at the molecular level in lots and lots of different species. We know that there's sex differences in the electrophysiology. I could go on and on at all the different levels of analysis. We know that they exist. We know that there are sex and gender biases in the phenotype and incidence of neurological disorders. I pulled this figure from a paper that Peg McCarthy wrote, right, which shows things like ADHD, for example, which is much more common in males compared to females, right? There's arguments about whether that's because it's easier to diagnose bias or something more biological, but it's still more prevalent in males based off of the available data. We have things like females where you see more prominent anxiety and depression, things like that, more likely to develop an addiction in response to a psychostimulant, a drug of abuse, things like that. We know that there are sex differences in not only normal behavior, but also in neurological disorders. And right about the time when I was on the job market, right, I'd finished my postdoc, as you're a graduate student, and then you go to your postdoc, and then hopefully at the end of your postdoc, you go on the job market, go find yourself a job, right? And for me, I wanted to become an academic scientist. I wanted to have the opportunity to run a laboratory, right? And to do that, you really have to make a pitch. I mean, you're asking a university to front you the money to run this thing, right? And so to me, that is a wonderful opportunity to think very, very hard about what type of question that you want to study. And not just what type of question, but where. In my case, where in the brain and in what animals, right? And so right around this time, this was in 2012 when I was on the job market, all this data was exploding across sex differences. Chris already mentioned I was working on sex differences in the hippocampus, okay? And this really led to a moment where I sat down and thought about, well, I know I want to study the role of biological sex, but like where? Okay, where? And so I thought, well, what characteristics should a brain region have to be an interesting place, not only to me, but hopefully other people, in which to study the intersecting roles of sex and hormones on neuron electrical properties. And so I decided, well, it should have clear estrogen and or sex differences in associated behaviors and disorders, right? and not just copulatory behavior or reproductive behavior, but lots of different behaviors, okay? Because I didn't want to just study a brain region that could be like dismissed as being yet another sort of sex-specific brain region, okay? I wanted that brain region to be relevant to a wide range of important functions, okay? I wanted that brain region to express membrane-associated estrogen receptors, which are a specific type of estrogen receptors that happen to be on the cutting edge of neuroendocrinology right now. I wanted that brain region to express aromatase, which is what transforms testosterone into estradiol. And then, most importantly, yeah, y'all read the slide, huh? Y'all see it in the back? I did not want it to have a bunch of senior neuroendocrinologists with big labs and lots of money studying it, okay? I wanted to be able to have a niche, at least starting out, right? So that meant that the hippocampus, the hypothalamus, and the song control regions were all out, okay? It was too crowded, okay? And so I hope, when I was invited to speak here, I was told that there were a lot of students in the audience. And so I really wanted to be honest with you about both the scientific and the practical ways that how I've decided to run our research program, okay? So y'all all know where I'm going with this, because in the title, you all know my favorite brain region, the striatum. And the striatum happens to meet every single one of those characteristics. So if we consider the striatum, here is in a schematic of a human brain that I pulled from an NIH talk. And so, if you think about a human brain, the striatum is buried deep within the telencephalon. If it's like my fist, cortex kind of wraps around it just like this, okay? And if we think about the rodent brain, which is schematized here, this is a parasagittal schematic of the rat brain. That means it's cut like this, okay? And so over here on this side, you have the olfactory bulb. Over here is the cerebellum. And then in the rodent brain, the striatum is always buried deep inside, right here, okay? And I'm not going to do a deep dive into striatal anatomy and physiology, although I'd love to do so. I could totally do it for the next hour, but I'm going to spare you, okay? And instead, I just want everybody to know two things about the striatum. Okay, number one, I want you to know that it's a big place, and it's divided into subregions, okay? And one subregion is called the dorsal striatum, also called the caudate-putamen, because I don't know about plant scientists or biomechanical engineers and all that, but neuroscientists, we love to have more than one name for the same thing, okay? I'm sure no one's ever encountered that, right? No one's ever encountered that. But we've got the dorsal striatum and the caudate-putamen. And this part of the striatum is hooked up into motor control circuits, most famously also habit learning, like all kinds of things like that, okay? And then the other part of the striatum is called the nucleus accumbens. It's also called ventral striatum because it's on the ventral side of the animal. It is further subdivided into the nucleus accumbens core and the shell. The part that's called the core is called the core because it wraps itself around that interior commissure, and the shell wraps around the core kind of like an eggshell, right? That's where those names come from. And so the nucleus accumbens core and shell are hooked up to the limbic system, among other things, and they mediate things like reward and motivation, okay? So if we think about disorders, right? So this area is most famously impacted in Parkinson's disease, that's the dorsal striatum, okay? And then the nucleus accumbens core and shell is most famously impacted in terms of depression and anxiety disorders and most prominently drugs of abuse, okay? All right, so that's the first thing. The second thing that I want you to know is even though there are all these different subregions and they're hooked up to different inputs and outputs, it's the same neuron types. Okay, so the most prominent neuron in the striatum is called the medium spiny neuron. It's my favorite neuron of all time. And the medium spiny neuron, okay, is about 90 to 95% of the neurons in the striatum. It depends on the species and how you measure them, but 90 to 95% of those neurons are medium spiny neurons. And then most importantly for me, they're also the projection neurons out of the region. Okay, and so the logic is that if sex and steroid sex hormones are going to modulate this region in any way, for that message to get out, the medium spiny neurons have to be affected, okay, in some way, right? So that targets our electrophysiological investigations to that neuron type. You may be wondering, I know I would be, is why we call them the medium spiny neuron. This is the kind of thing that keeps me up at night. Why do they call it that, right? And they call it that because the soma of the cell is medium in size compared to other neurons. And then the dendrites, the part of the neuron that receives information from other neurons, is filled with these little protrusions these beautiful little protrusions. I think they're cute little protrusions are called spines. And they're the site of excitatory synaptic input onto these cells, among other places. Here they are blown up. This is a Golgi stain that Opal and Laura did in my laboratory, two very talented undergraduates. And you can see these little spines coming out. And then those spines receive excitatory synaptic input. They're going to also be excitatory synaptic input other places, but most prominently associated with the spines. Okay? And so the median spiny neurons, they exclusively express these membrane-associated estrogen receptors, both ER alpha, ER beta, and GPAR1. We see very little nuclear localized ER alpha in the striatum, but if someone like Ann Almy did an electron microscopy experiment when she was still in Wayne Brake's lab, you see these beautiful little labels in the dendritic spines and the axons and the soma and the dendrites of these cells. And then if we look at a different type of estrogen membrane estrogen receptor like GPAR1, Ashlyn Johnson and Christian Korenschnabel in my laboratory did that staining, and we see lots of nice puncta. So we know that estrogen receptors are there. We know that it expresses aromatase, at least in rats, mice, sheep, and human. Those are the species that it's been investigated in using both qPCR and immunocytochemistry. Here's the nucleus accumbens core right there. And then we know that the striatum, actually we've known for decades and decades, since at least the 70s, and maybe even earlier, that the behaviors and disorders associated with this brain region show sex differences and sensitivity to estrogen, okay? And the striatum is a big place. It regulates a lot of different things. So not surprisingly, we see sex differences or estrogen effects in striatal-mediated learning tasks. We see sex differences or estrogen effects in impulsivity, in sensorimotor locomotor behaviors and tasks, in reproductive motivation and pacing mating behavior, in parental behavior, in some aspects of Parkinson's disease, especially L-DOPA-induced dyskinesia, and then of course in multiple aspects of addiction, okay? And so to kind of give you a flavor of this literature, this is a classic experiment that Jill Becker did way back in the 80s, published in 1987. She did a really simple behavior task. She gave a rat the opportunity to walk across a balance beam, and she just counted how many times it screwed up, okay? How many times that it fell off the balance beam. And so one of the things that she did is she took female rats and she tracked their estrous cycle, remember, because those estrogen and other hormones, they fluctuate, right? And so depending on the estrous cycle, the number of foot faults on average changed across the estrogen cycle. And then when she took a gonadectomized rat, which means that she took out the ovary, right? And then she implanted estrogen into the dorsal striatum, we see this very nice reduction of footfalls, and then it comes back up again, right? And this is sort of stuff has been replicated since then, but we know that there's a behavioral differences. And so, naively, you would kind of expect if, we've known about these behavioral changes.
Sorry.
Please.
Please go back to this one.
Mm-hmm.
So these were all rats that they were same age?
Yes, adult.
And same gender, I assume.
Yes, they're all adult female rats.
Okay.
That's a very important question and the reason why it's a very important question is because we know that the contributions of steroid hormones differ across development, right? And we know, at least in some locomotor tasks, especially in open field behavior, that some of the sex differences that you see don't emerge until after puberty, right? But then other things like impulsivity, right, emerge prior to puberty, okay? All right, so with all this behavioral data, naively, I would sort of expect that, well, it's been known since the 70s, and it should be all figured out, right? It should be all figured out. And, and there has been a lot of work, all right? There's been a lot of work, and it is focused on things like gene expression, transcription factors like CREB and CFOS and delta FOSB, changes in dendritic spine density, and especially in dopamine action. There is a large and robust literature on estrogen modulating dopamine action and release of all sorts. I'm going to reference that a little later. Okay, but in terms of the electrophysiology of these neurons, prior to the establishment of our lab, there were only 2 studies, at least that I know about, 2 studies. Okay, and one showed that estrogen modulated L-type calcium channel currents, and the other showed that there was a sex differences in adult rats in the number of excitatory synapses in the nucleus accumbens core, favoring females compared to males, okay? And the reason why I care so much about the electrophysiology of the neurons is that that's the output, okay? And that all the changes in gene expression and molecules and everything else, if it doesn't change the way that the neuron integrates and produces an action potential, it doesn't affect the next neuron in the circuit, and it can't alter behavioral output of the animal, okay? So I like to study those action potentials because I can make arguments about how it actually changes the dynamics of those groups of neurons that changes behavior. Plus, I like looking at action potentials, okay? All right, so back in 2013, I took these two studies, and I took everything up there, and I made my first table, okay? I made a table of everything that we knew about regional sex differences and similarities in medium spiny neuron electrophysiology across development, okay? And the table is pretty simple. On this column is broadly speaking, the electrophysiological property, and I've divided that up between intrinsic excitability, which means things like, that means stuff like if an ion channel expression changes in the neuron, or how the neuron fires action potentials, things like that, okay? And then the other way I divide it up is excitatory synaptic input, right? So how those synapses function, right, onto the medium spiny neurons, okay? And then I divided it up by developmental stage, so a very gross division of developmental stage, mainly pre-puberty versus post-puberty. And then I divided it up by striatal region, so dorsal striatum, the nucleus accumbens core, and the nucleus accumbens shell. And here's what was known, right? And so when I made this graph, what I saw was opportunity, okay? Opportunity, because there were lots and lots and lots of question marks of things that we didn't know, okay? And that's exactly what you want when you're starting a lab, okay? And so there were lots of question marks, but we knew from that earlier literature, especially from the dopamine literature, that dopamine, right, exposure release was different in females compared to males. Generally speaking, females and estrogen can increase dopamine action compared to male, okay? There's a lot of subtleties in that, so I don't want you to push that analogy too hard. But for example, people had done electrochemistry and microdialysis and other things to show increased dopamine released and retention in the striatum in females compared to males. And the reason why that is important is because this molecule dopamine modulates medium spiny neuron excitability, at least in males, okay? And it can modulate not only excitability from an intrinsic perspective by acting directly on the medium spiny neuron, but it can also modulate excitatory synaptic input, both sort of on the short-term and the long-term scale. And so based off of this data, I was able to make a prediction that sex differences exist in either or both medium spiny neuron excitability and excitatory synaptic input. And once I had made this prediction, I was then able to refine that into hypotheses. And so broad hypotheses like sex differences exist in medium spiny neuron cellular excitability, that they exist in medium spiny neuron excitatory synaptic input, and that the sex differences vary by striatal brain region and by developmental period. And now I'm going to address these hypotheses in turn in each striatal region, the dorsal striatum, the nucleus accumbens shell, and the nucleus accumbens core. And the way that we're going to test these hypotheses is we're going to use a technique called the acute brain slice and whole cell patch clamp, okay? So what an acute brain slice is we take an animal, either a prepubertal animal or an adult animal, we anesthetize the animal, deeply anesthetize the animal, and then we rapidly extract that brain, and then we slice it up into really thin sections. So in our hands, we usually use a 300 micron section of brain, okay? And once we have that section of brain in the brain region that we care about, right? So we can take a section right through the dorsal striatum. We can take that brain slice and we can keep it alive. In our labs, we usually keep it alive 4 to 5 hours. Some people will say longer, some people will say shorter. For us, it's 4 to 5 hours. And we can keep it alive in oxygenated artificial cerebrospinal fluid, okay, that matches what the brain would naturally experience. So once we have that brain slice, we can put it on a microscope. So here's the slice on the microscope. Here's a little harp that keeps that in place. It's sitting in a pool of oxygenated artificial cerebrospinal fluid. The striatum is right about there. Here's the objective from the microscope. And we can lower that objective down and then use IRDIC microscopy. So that's infrared light because it passes through tissue very easily. And differential interference contrast is just a fancy name for making shadows appear in the tissue. okay? And so on lower power, it looks like this. Here's all the little dots. Those are living neurons. And if we increase the magnification, here's a living neuron right there. That's about 15 microns in diameter. So we can visualize the neurons. And once we see the neurons, we can then target them with a glass electrode that's filled with a solution that both conducts electricity and doesn't mess up the cytosol of the cell very much, right? Because those of you who are cellular biologists are going to know that if you mess up the cytoplasm, you like alter cellular function, right? So we slowly lower that electrode down through the tissue. The tip of this electrode is about a micron in diameter. We're exerting positive pressure the entire way so this thing doesn't get clogged. When we get next to the plasma membrane, we release the positive pressure, we apply negative pressure, and it seals the tip of that electrode onto the plasma membrane of the cell. And then we open up a tiny hole in between the inside of this electrode and the inside of that cell, and that's what gives us electrical access to the neuron. And once we have that access, we can do two things. We can both record what the neuron is doing, and then we can ask it questions, right? And the way that we ask it questions is by injecting like electricity into it, right? Because neurons operate as electrical cells, right? And so what that looks like is this. So this is a screenshot of one of our experiments. Then the y-axis is millivolts. On the x-axis is time, and each one of these columns is a second. So these experiments are very dynamic. The data rolls out at you at real time, okay? And so we can do things like the neuron is resting here, and then we can inject a little bit of positive current to mimic an excitatory synaptic input, and that depolarizes a little bit, and it comes back down again, and then we can inject some more, and then boom, there's the action potential. The most beautiful thing in all of biology, okay? I get the stage so I can say stuff like that today. No, you'll forgive me later, right? So we have an action potential, and then it comes back down again. We inject a little more current, it fires some more action potentials. We inject a little more current, it fires some more action potentials. So my technician and I, David Doris, we did that. We did just that experiment in male neurons and female neurons from prepubertal dorsal striatum. It was one of the first experiments that we did in my lab, okay? And what we noticed right away from just looking at the traces that there were some subtle and some not so subtle differences on average in the electrical properties of male and female neurons. And so if we take males on the left, females on the right, here's where we injected just a little bit of current, just enough to induce an action potential. You'll notice that this after hyperpolarization is a little smaller than this one, right? If you inject a little more, the male fires maybe 5 action potentials, the female is firing 6, right? And that's a small change, but on average, across many different neurons, that leads to a real change in excitability, okay? And so at this point in the talk, I could follow up with a really wonky electrophysiology talk, where I show you all the experiments and controls and analyses and everything else. And I'm going to spare you, okay? I'm going to spare you. You can thank me later, okay? And I'm happy to talk shop about electrophys in the Q&A if you want, okay? And instead of talking about all the different analyses and everything else, I'm going to focus on one metric, which shows very clearly the change in excitability between males and females, because we see an increased amount of excitability in those female neurons compared to the males. And the reason why I can say that is because these females are generating more action potentials per the same amount of current than the males, okay? And so the way that I'm going to show you to illustrate this as a metric is a metric called the FI slope, which is an acronym for the slope of the frequency of evoked action potentials per injected unit current curve. Okay, and that's a mouthful, okay? But all it is, I want you to just go back to basic geometry and think slope, okay? And so what you have in front of you is a graph, and on this axis is action potential frequency. So this is the number of action potentials evoked in hertz, so that's evoked per second, right? Versus the amount of positive injected current. So medium spiny neurons, they have a very hyperpolarized resting potential. So you start injecting current and nothing much happens, and then all of a sudden, boom, there's your first action potential. You inject a little more current, and the action potential number increases, and in medium spiny neurons, eventually they plateau, right? And so one popular way of showing, of course I just like turn this off, here we are, of showing an excitability of a medium spiny neuron is just to set a slope line between here and here, okay? And if there's a steeper slope, that means that that neuron is more excitable, and if it's a shallower slope, that means that it's less excitable. Okay? You can analyze this lots of different ways. You can fit curves. You can do all sorts of things. A very gifted mathematician, Lindsay Coons, joined the lab and analyzed this a different way and found exactly the same thing. So I feel pretty confident in it. And if we do this type of analysis, we see this increase in excitability in females compared to males. This is a bar graph showing you the means. And if we look at the distribution, so each one of these is an individual cell, we see a very nice shift in the distribution, right? But what you'll also notice from this is that there's overlap between males and females, right? So this is a sex difference. It is not a sexual dimorphism. It is a sex difference, favoring increased excitability in the females. So we were overjoyed with this, and we started doing a number of different experiments. We don't know the complete mechanism yet, but we've been, of course, focusing on ion channels. And if we do experiments like PCR experiments and measure things like voltage-gated sodium channel subunits, in some of them, we see an interesting increase in females compared to males. This research is ongoing, but most importantly for me, I was able to go back to my table and I was able to fill in the first blank. And that's a real victory for a new lab, y'all, your first experiment, okay? And so just as there's the intrinsic excitability, right? So that's the integration that neurons do inside of the membrane. There's also all these inputs coming in. So in order to interpret this and know whether it's a real increase in excitability or if it's some sort of homeostatic adaptation, you also have to assess the excitatory synaptic input, okay? And one way of doing this is to take that neuron and to put it into a technique called voltage clamp and to apply drugs that block GABAergic synapse and action potentials and measure a thing called miniature excitatory postsynaptic currents. And if you do that experiment right, what the data looks like are these cute little blips. Blip, 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 right? And what you're seeing with each one of those blips is the response of the neuron to a spontaneously released quanta of neurotransmitter from the presynaptic side, okay? So we're picking up how the excitatory synaptic inputs are talking to this neuron. And there's a number of different analyses that you can do for this. The one that I'm gonna focus on is the frequency of these. And that's simple, you just count how many of them are per unit time. And if there are differences in the frequencies of these things between, say, like males and females, that usually tells you things about the presynaptic side. That can say, well, maybe there's a difference in excitatory synapse number between males and females, or maybe there's a difference in the machinery of the release probability of that, okay? So we did this experiment, and initially we were really excited because we got these two data points first, and we were like, woohoo, it's like this huge sex difference, right? And then we went back and did a bunch more experiments, and they all like overlapped, right? And so that leads to a very interesting graph that looks like this, which is not statistically significant whatsoever. And so from this, I conclude that the preponderance of evidence suggests that there is no robust sex difference in excitatory synaptic input in dorsal striatum, at least so far. Okay, so if we go back to our graph, I can now fill in here. Right? And so what this data argues so far is that this increased intrinsic excitability is an actual real increase, and as far as we can tell, it is not a homeostatic adaptation to sort of like bring the sexes closer together, but is actually an increase in excitability.
I actually think the fact that there is no difference it doesn't really surprise me. I wouldn't expect that you will see difference in all the different parts of tissues that you know.
I agree. And the reason why...
Suspicious of something like that.
Absolutely. I would be suspicious too. And the reason why I think both of us would be suspicious is that we're really talking about a sex difference here, and there's a lot of overlap between males and females. So a priori, we would expect a lot of sex similarities, and then the differences that exist would be shifts in distributions. Okay, obviously I can't speak for you, but I can speak for me, right? And so the other reason why this is important is because if we take a neuron, hopefully I can draw on this thing, right? We take a neuron, and if there's an increase in excitability, okay, that's great. It makes like an action potential, right? But let's say that the number of excitatory synaptic inputs was actually decreased in females compared to males, right? And if that would have been the case, then this increase in excitability that we would have measured functionally would probably not have been an increase in excitability at all. Okay? So that's why you have to do this kind of experiment. It's like a yin and a yang experiment. Okay? And of course, there's inhibitory synaptic input as well. We haven't gotten to that yet. All right, so if the pointer would work. Oh, no. Nope. All right. So the other thing that matters with this is that so far we've only worked in prepubertal animals, but I made a big point of saying that this neuroendocrine environment is dynamic, right? And of course there are differences in cycles in adult females, right? And so in humans we have the follicular and the luteal phase, and then in rats we have these different phases of the estrous cycle, sometimes when estrogen levels are high, like in proestrus, right? And so we know this is important for the striatum because there's been decades of research showing cycle-dependent sex differences in striatal behaviors, including things like increased locomotor activity. Female rodents run like crazy during proestrus, okay? And there's other differences in behavior as well. So my graduate student, Jamie Willett, took that data and formulated the hypothesis that the electrical properties of these dorsal striatal neurons are potentially altered by the cyclical hormone changes of the estrous cycle. And she mapped these neurons across the different phases, and we see a beautiful increase in excitability during proestrus compared to diestrus when estrogen levels are low and compared to males. Okay, so this argues that the excitability data that we see is not only different before puberty, but is also different between not just within females, but females compared to males during proestrus, at least parts of the estrous cycle. So now we can begin to fill in this graph, but what about the other brain regions, right? So there's 3 brain regions. We just did this, so now we're gonna do the exact same experiments as before, except we're gonna look at the shell. Jamie Willett also did these experiments, and she was super excited to start this project, and she was disappointed by the end of it because she looked really hard, and she found no sex differences whatsoever in these control animals. And so I should note these are control animals, and I'm sure Dr. Hodes and others would have lots to say about environmental influences that can push sex, that can create or push sex differences one way or the other. Okay, but at least in the control animals, she didn't see any differences in either intrinsic excitability or in the properties of the excitatory synaptic inputs. So if we return to our table, now we can begin to fill in these blocks, and then we can do the exact same experiments again, this time in nucleus accumbens core. These experiments were done by Dr. Jinyan Chow in my laboratory. And in nucleus accumbens core, it's a different story. We see sex differences, but not in the intrinsic excitability of these neurons, but in the excitatory synaptic input. So here are those traces that I showed you before with the little blips, right? And each one of these blips is an excitatory glutamatergic synapse talking to the neuron, right? And so in males, there's fewer blips than in females. There's a lot more blips in females. And if we analyze that, there is almost a 2-fold increase in excitatory synaptic input as indicated by miniature excitatory postsynaptic current frequency in females compared to males. And here's the distribution here, okay? And so this difference in excitatory synaptic input is present pre-puberty, right? And so that argues that it's present long before adulthood, right? But Stephanie Proagno in my laboratory has been recording these same cells in adults, and she's finding that there are significant differences across the estrous cycle in the amount of excitatory synaptic input onto the core. So here's diestrus when estrogen levels are low. Here's proestrus when they're high. Estrus is the stage of the cycle right after proestrus. They begin to fall. And these are animals where she removed the ovary. Okay, and they have an intermediate phenotype between di and estrus. So we know at this point that there is a sex-specific programming of excitatory synaptic input in the nucleus accumbens core that occurs long before puberty, and then after puberty that these inputs are regulated by the estrous cycle. Okay? And so now we've begun to fill in this graph, right? And obviously we still have some holes to fill, but we've built our sandbox, okay? And we now have a foundation that we can do interesting experiments about mechanism. How is this converted? Why, right? Both at the level of the endocrinology, at the level of the ion channels, at the level of whichever particular variable you happen to be interested in, right? And so one of the first experiments that we did to study mechanism is to figure out what is the role of hormones in programming this sex difference, okay? And in order to do that, right, we drew heavily on the data that was generated by those historical models that I mentioned at the very beginning of my lecture, right? And we knew from those models that things that drive neurons into a more phenotypically masculine or feminine state include things like genetic chromosomal effects, such as XX or XY. We know that steroid sex hormones acting during those sensitive critical periods can permanently modulate those structure. And of course, things like experience, environmental inputs, sociocultural influences, epigenetic influences, right, can all modulate these things. And so we decided to focus on whether steroid sex hormones programmed this sex difference. And we returned to that findings, right, that the male rats experience a masculinizing testosterone surge soon after birth. Here's the data actually plotted here, replotted from one of Ward's papers. Here's testosterone, plasma testosterone. Here's hours after delivery. Here's that peak in males, and here's the baseline in females, right? And that leads to the hypothesis that early hormone exposure in these males decreases mini EPSC frequency. And if that hypothesis is correct, then we should be able to take female animals and inject them with either testosterone or its metabolite estradiol and mimic the effect. We should be able to generate a male effect, okay, if they're injected during this sensitive critical period, okay? And so Dr. Jin-Yen Chow did this experiment in the lab. And the way that she did it is that she took rats and she injected them with either vehicle solution or testosterone or estradiol on the day of birth, the day after birth, and the second day of birth. We waited for the animals to grow up to prepuberty, so then we recorded them between P16 and P23. And what she found is that both testosterone and estradiol blocked this increase in excitatory synaptic input. So here's the vehicle here showing the sex difference, and then here's males and females both exposed to estradiol, and the sex difference is obliterated, and then here's the exposure of the females to testosterone, okay?
Sorry, and the receptors are expressed at the same level throughout that, I mean, I assume that you check different points over
Yeah, so we've checked at specific points, but we haven't checked, and no one has checked, all the way through that entire neonatal period to say with confidence that expression levels between males and females is like the same or different throughout that level. We know that they're there, but we don't know if they change through that time. You want to come work in my lab? You got a lot of great questions. Sabbatical, you know. So what this argues is that early hormone exposure is programming sex-specific excitatory synaptic input into the nucleus accumbens core onto medium spiny neurons only in the nucleus accumbens core, okay? And so this is obviously important from a neuroendocrine perspective, but this also opens up interesting implications for environmental stimuli that also occur in and around birth, at least in rodents. And so all this data that I've shown you has been generated over the last 4 years. And it allows my laboratory to now center around the following central hypothesis that's driving our work now, right? Which is in early development, right? So that means before puberty, right? And we know that because of all the prepubertal analyses that we've done. Estradiol, right, which we know from the experiment that I just showed you, regionally programs medium spiny neuron electrical properties. And so regional because you don't see the same sex differences and similarities across all the different striatal regions, okay? And then in adulthood, temporarily modulates them via sex-specific membrane-initiated mechanisms. Temporary modulation is from the estrous cycle experiments. Sex-specific is because the females have estrous cycles and the males do not. And membrane-initiated, we don't have any evidence for that yet, but that's why you build a model, right? So now we can test it, okay? And so we have our central hypothesis. And so to summarize really the data portion of this talk, What I've shown you is data that indicates that medium spiny neuron electrical properties show sex differences in some respects, sex similarities in others, in multiple striatal regions, but that they're different. So that in the dorsal striatum, we see a sex difference in excitability, favoring females over males. This is modulated during the estrous cycle. In the core, we see sex differences in excitatory synaptic input, created long before adulthood and puberty, sensitive to early exposure to estrogen and testosterone, and it's modulated in adulthood by the estrous cycle. And at least in prepubertal animals, we don't see any sex differences in any measurement between males and females, okay? And so that's the baseline data, and now I'm gonna speculate a little bit, okay? So bear with me, I'm gonna speculate for a few minutes on perhaps some broader implications of this, okay? And so, if we were not to consider sex at all, right? If we were just to sort of ignore sex and just treat the brain as sort of this, unsexually differentiated object, right? That's obviously not supported by our data, right? Our data argue that at least in some properties and some brain regions, we see an effect of biological sex, okay? So that really doesn't work for me, okay? And so let's add on a level of sophistication, right? And so sometimes you see people argue things like there's a dimorphic brain, there's a male brain, and there's a female brain, okay? And this is really a very simplistic view, and I think usually when we start making broad generalizations like that and very simple generalizations like that, they're usually wrong, right? And so the data that I showed you today in the shell, right, at least at the time point that we considered, we didn't see any sex differences, right? But in the core we do in one particular aspect, and in the dorsal striatum we do in one particular aspect, and then there is quite a bit of overlap between the males and the females, right? It's a difference, right? There's differences in the tells, but there's overlap in between. Right? So to me, that argues that we need to be very, very, very careful about making stereotyped generalizations between male and female brains at an entire level, okay? And so now what I'm really thinking about is the brain as more of a mosaic, okay? And I put a question mark there because I'm not sure if that word is right just yet. But I'm thinking of it in terms of a mosaic. Let's just call it a complex view. Right, with each brain region showing differential masculinization and feminization, right, depending on what sort of estrogen receptors are there or environmental inputs and things like that. And I wonder that as we continue to study individual neurons, right, that even we become finer and finer in this, where there are particular neurons that are more or less susceptible to sexual differentiation. And that really, one of the reasons why we have this large variation, not only in neuron properties, but also behavior, right, is because we have all sorts of influences, including biological sex and other variables, which craft and help differentiate neuron function to push the systems and tweak them just a little bit, do something just a little bit different, okay? And so I'm speculating, okay? I'm speculating here. And one of the reasons why I know that I'm speculating is that in most systems meaning parts of the brain and neuron types in the brain and glial types in the brain, like we don't know when sex matters and when it doesn't, okay? Because people haven't done the experiments. So let's think about just the medium spiny. How am I doing on time? Am I good? All right, I got 2 minutes, okay. So let's just consider medium spiny neurons for a second. Well, 2 minutes, okay? So medium spiny neurons, okay, they're one of the most studied neurons in the history of neuroscience, okay? People have been patching medium spiny neurons for a long, long time. And one of the things that astounds me about not only my work, but all the other people doing such wonderful work with the effects of sex and steroids on the basal ganglia system is why we're just discovering this now. Okay? And so, the naive thing is, oh, maybe the techniques weren't present, right? And so I'll tell you that all the techniques that I showed you today, let's think about patch clamping. That's how we were able to measure the intrinsic excitability and the excitatory synaptic input and all that. Sackman and Nair invented patch clamping in the 70s, okay? And the first whole cell patch clamp with a gigaseal, which is a technique that we use, was published in '81 by Hamill. The first sharp recording, which is an alternative technique, was done by Eunice, published in 1980, okay? So patch clamping has been around a long time. Okay, what about the brain slice preparation? Okay, that brain slice preparation, it was invented and first published in the 50s, 1951 by McElwain. He was a biochemist, and he invented it because he wanted to study brain like biochemistry. Okay, and then it was the first published methods for applying it to patch clamping was in 1989. Of course, it was used before that, but they didn't publish the methods. Okay, you can speculate yourselves why. But 1989, first published methods, okay? And then the discovery of males and females, okay? I mean, we don't know when would have been a good day though, okay? Would have been a good day. Earliest known icon, right, of a earliest known sexually differentiated icon is 35 to 40,000 years old, the so-called Venus icons. That's a problematic name. But I looked for documentation, y'all. That's the oldest documentation I could find of that, okay? So my point is that the reason why this wasn't discovered is because nobody asked the question, okay? And it is a simple question, right? No one asked the question. And that is not just specific to medium spiny neurons in the dorsal striatum, but it is documented across neuroscience. So Bury and Zucker and others have done a very nice job at documenting this, and that neuroscience and other fields, to be fair, we have a history of either not reporting sex at all, it's called sex omission, or favoring the use of male animals over female animals, okay? And that obviously is a complicated subject and stirred great and fierce and passionate debate within our community, okay? But we know that these phenomenons persist in that I formed a team of 11 curators who sorted through over 10,000 neuroscience articles published in Nature, Science, Neuron, Nature Neuroscience, J Neuroscience, J Neurophys, okay? And then they just further analyzed the ones that use rats or mice, okay? And that was over 6,000 articles. And what they found, there's good news and there's bad news, okay? And I'm an optimist, so I'm gonna give you the good news first. And the good news is that as of 2010, which is after Bury and Zucker's paper, okay, over 40% of neuroscience articles were not reporting sex at all, but by 2014, this had dropped to 20%. So that's excellent progress, okay? And we can talk about whether 20% is like an acceptable number. I would argue it's not, but it's still a wonderful increase, okay? And that the percentage of increase of articles using both males and females has been increasing. The number of males is pretty steady, but going up. And here's the number of articles that analyze just females, okay? And so these articles that study males and females, here's the bad news, y'all. Only about 20-25% of them provide any kind of documentation that sex was used as a biological variable. And we use a very liberal definition of that, okay? We know that this phenomenon varies between mice and rats. It varies between animal species. And we know that it varies quite a bit by journal, okay? So for example, the Journal of Neuroscience showed a very large drop in the percentage of articles that are not reporting sex. And as of 2014, there were some other journals that were still pretty high, okay? And so I don't argue, unlike some of my more radical colleagues, I don't argue and I don't believe that everybody has to necessarily study both males and females. But what I do think is that you should report the sex of the animal that you study, okay? And when you choose the sex of the animal that you study, you should be thoughtful about it and use it for a reason and recognize that things may be operating a little different in other circumstances, okay? And so with that, I will get off my soapbox and I will thank the members of my laboratory who have produced almost all the data that I showed you today. I'll thank the Grass Foundation, the Army Research Laboratory, and the National Institute of Mental Health for funding our work. And I thank all of you for hanging with me through this talk. Thank you very much.